Good evening, everyone.
Good evening, sir.
So this is 8 o'clock. Am I correct? So this 8 o'clock. Correct. Let's exercise a little patience. Let's see uh, how far with people. Let's set some patience so we can actually have more people to the. Let's exercise a little patience. Let's see uh, how far with people. Let's set some patience so we can actually have more people. Let's exercise a little patience. Let's see how far with people. So let's see if we can actually give some time so that more people can join. Before we proceed, we have up to 5 persons streaming live now. Let's see if we can give a grace period of 8:15. This is just. Let's see if we can actually give some time so that more people can join. Before we proceed, we have up to 5% streamline arm. Let's see if we can give a grace period of 8:15. This is just activated for time. We have about let's say 6 minutes more. Then we'll start. So we have about let's say 6 minutes more. Then we'll start. So we have about let's say okay. Okay, so you can actually hear me now, right? Okay, so you can actually hear me now, right? Okay, let's proceed with the lecture. The video will be posted to YouTube after this lecture so that people can actually have and view again. So my name is Raymond Ibe. I'm a lecturer. Okay, let's proceed with the lecture. I'm a lecturer of the Federal University of Technology, Owerri. The video will be posted to YouTube after this lecture so that people can actually have view again. My name is Raymond Ibe. Lecturer, lecturer of the Federal University of Technology, Owerri. So our learning objective for today's lecture will be focused on this major content. So we're looking at amino acid, how we can identify the 20 amino acids that are found in proteins. So our learning objective for today's lecture will be focused on this major content. We're looking at amino acids, how we can identify the 20 amino acids that are found in proteins. We're also looking at how we can actually draw the structure of the 20 amino acids for students of biochemistry and how you can identify the structural difference between the 20 amino acids that are found in proteins. For students of biochemistry and how you can identify the structural difference between the amino acids that are found in proteins. We'll also be looking at the essential and the non-essential amino acids. We'll also be looking at the essential and the non-essential amino acids. What makes them essential and what makes them non-essential? The arrow group rulers, which are actually used to classify the amino acids based on their characteristics. The 20 amino acids, what makes them non-essential? The 20 amino acids that are found in proteins the amino acids based on characteristics. 20 amino acids, the 20 amino acids that are found in proteins. Based on characteristics. 20 amino acids. So we are looking at the protein placement where we can actually predict the location of an amino acid. For example, if an amino acid is found in a protein, where likely will the amino acid be found in that protein? And this is actually determined by the characteristics of the amino acid. Location of an amino acid. For example, if an amino acid is found in a protein, where likely will the amino acid be found in that protein? And this is determined by the characteristics of the amino acid. So we also look at the, we can actually, at the end of this lecture, you guys will be able to draw the structure of a protein irrespective of the chain length. So we also look at the, we can actually, at this lecture, you guys will be able to draw the structure of a protein irrespective of the chain length. So we also look at the, actually, at this lecture, so let's look at amino acids. Now, amino acids are actually a group of biomolecules that have these three distinctive features, more especially the ones that are found in proteins. Let's look at amino acids. So if you look at this, amino acids are actually a group of biomolecules that have these three distinctive features. One, especially the ones that are found in proteins. One, they must have the carboxylic acid. That is where, that's actually one of the, how they coined the name amino acids. Two, they must have an amino group. So the amino group with the acidic group, which is the carboxylic group, makes it amino acid. That's actually one of the, how they coined the name amino acid. That we have the, two, they must have an amino group. The hydrogen. The amino group, the acidic carboxylic group, amino. Now, these three features, the carboxylic group, the amino group, and the hydrogen, is unique to every amino acid that is found in protein, except proline. But when we get to proline, I will explain further why proline is referred to an amino acid. Proline is the only amino acid that have a different name. Others are called amino acids, but for proline, it is called an amino acid. It's referred to an amino acid. Instead of amino, it will be called amino acid. That have a different name. Others are called amino acids, but for proline, it is called an amino acid. Now, let me take you down, let me take you a little to the nomenclature or the numbering of the carbon atoms in amino acids. You know, in nomenclature, when we were taught nomenclature of carboxylic acid, we were taught that the carbon after the carboxylic group is referred to as the alpha carbon. In amino acid. In nomenclature, when we were taught of carboxylic acid, the alpha carbon is where the amino group is attached to. That is why they call them alpha amino acid. The alpha carbon is where the amino group is attached to. That is why they call them alpha amino acid. You can see the alpha carbon here. You can see that this is the alpha carbon. This is the alpha carbon. You can see the alpha carbon here. So the amino group is attached to the alpha carbon. That is why it is referred to alpha amino acid. There are other amino acids that are not alpha amino acid. We have gamma aminobutyric acid, which is GABA, a neurotransmitter. It's an amino acid, but it is not an alpha amino acid. So you might be asked the question, which of these amino acid is not found in protein? The amino acids that are found in proteins are called alpha amino acid. So if it is not alpha amino acid, it cannot be found in proteins. Which of these amino acids is not found in protein? The amino acids that are found in proteins are called alpha amino acids. So you might see something like gamma butyric acid, which is GABA. It's an amino acid, but not an alpha amino acid. It's actually a neurotransmitter. So the alpha carbon atoms in amino acids are chiral, except for glycine. Glycine is the only amino acid that is optically inactive. In the next slide, I will show you what is optical activity. Now you can see that. Well, how do we describe a chiral center or an asymmetric center? A chiral center is a center that has 4 different groups or functional groups attached to a particular carbon atom. Now, if you look at this, for glycine, we have amino group, we have carboxylic group, that is 2 now. Then we have 2 hydrogen and 2 hydrogen here. So you can see that 2 are the same, 2 are different. But for we to have a chiral center, the 4 of them must be different. This is like what we have in aspartic acid. You can see the amino group 1, the carboxylic group 2, hydrogen 3, then this other group here 4. So this carbon atom now is a chiral center. So this molecule will be optically active. It will be optically active. So that's why we say sometimes you'll be asked a question that which of these amino acid is optically inactive? If you see glycine there, glycine is definitely the answer. Glycine doesn't have a chiral center. That is why it is optically inactive. You can see phenylalanine. You can see phenylalanine. This is 1. You see glycine there. Glycine is definitely 2. Glycine doesn't have chiral center. That is why it's not 3. And a phenyl group, making it what? 4. So this carbon atom becomes a chiral center. So to rotate a plane of polarized light to the right or to the left. Now let me show you what is the plane polarized light. 3 and the phenyl group making it what? 4. So this carbon atom becomes the chiral center. So to rotate a plane of polarized light to the right or to the left. Now let me show you what is the plane polarized light. You can see the video playing. Here is a sample tube where that contains your sample, either glucose, each of the amino acid, aspartate, phenylalanine. So that is where the sample will contain. That is the sample container. This is the light. You can see the beam of light, which is called the unpolarized light. So the polarizer will make sure that a particular wavelength of light, a particular light path is seen. You can see the polarized light. So if the molecule inside this sample container, you can see the light going, changing direction. You can see it is plus 45 degrees. So make sure that a particular wavelength of light, a particular light path. So you notice that you can see the polarized light. So if the molecule inside this sample container, you can see the light going, changing direction. You can see it is plus 45 degrees. So you notice that please, you can drop some comment on the chat column. Let me see how we are progressing. Let me see how we are progressing. So please, you can drop some comment on the chat column. Let me see how we are progressing. So when we were discussing about the plane of polarized light, I know some of you see it as a fiction. What is plane of polarized light? How the molecules rotate the polarized light, either to the left or to the right. If it rotates to the right, it is called dextro-rotatory. Dextro, which is D-E-X-T-R-O, dextro-rotatory. If it rotates to the left, it is level rotatory. So that is why you see some molecule having some prefix before the name. You will see D or you see L. If you see D, you can see something like D-glucose, L-glucose, D-alanine, L-alanine. So whenever you see the D as a prefix, it means it is dextro-rotatory. It will rotate that light to the right. But if it is L, you know that it will rotate the polarized light to the left. But if it doesn't rotate the polarized light at all, you know that the molecule is optically inactive. So whenever you see the D as a prefix, it means it is dextro-rotatory. It will rotate that light to the right. But if it is L, you know that it will rotate the polarized light to the left. But if it doesn't rotate the polarized light at all, you know that the molecule is optically inactive. I want to see your comment. I want to see your comment on the chat column. Let me see how we are progressing. I want to see your comment. I want to see your comment on the chat column. Let me see how we are progressing. And how many of you enjoyed that little video of polarimeter showing how the molecule can rotate the plane of rotate the light either to the right or to the left? I don't know if you enjoyed that video. I know it has been a fiction when you hear something that they say it is dextro rotatory or level rotatory. How many of you enjoyed that little video of polarimeter showing how the molecule can rotate the plane of rotate the light either to the right or to the left? You enjoyed that video. I don't know if you done. When you hear something that they say destro rotatory or level rotatory. Okay, somebody asked the question, how can we differentiate between polar and non-polar amino acid? Of course, I will answer the question. Just keep tabs with me. I will answer the question. But how many of you enjoyed that? How many of you enjoyed that video of polarimeter showing how the molecule was able to rotate the light to plus 45 degrees? Somebody asked the question, how can we differentiate between polar and non-polar amino acid? Of course, I will answer the question. Just keep tabs with me. I will answer the question. But how many of you enjoyed that? How many of you enjoyed that video of polarimeter showing how the molecule was able to rotate the light to plus 45 degrees? Someone asked, how can we differentiate between polar and non-polar amino acid? Of course, I will answer the question. Just Now, this is non-polar aliphatic arrow groups. You know, I told you that it is the arrow groups that differentiate every amino acid. For example, the 20 amino acids that are found in protein, it is the arrow group that differentiate all of them. So they are classified based on their characteristics of the arrow group. Now, the arrow group is what you're seeing painted. Now, you noticed earlier that I said this is non-polar aliphatic arrow group. So I told you that it is the arrow group that differentiates every amino acid. I said every amino acid must have this one, the carboxylic group, the amino group, the hydrogen. These three are constant for the amino acids. The only group that differentiates the amino acid is this group here, which is called the arrow group. So if hydrogen is placed there, we call it glycine. If a methyl group is placed there, we call it what? Alanine. We call it alanine. So remember, I told us that proline is the only amino acid that has the name amino acid. The reason why it's called an amino acid, because every other amino acid, the amino group is actually a primary amino group. While for proline, it is a secondary amino group called an amino group. So it is called amino acid. Secondary because we have two bonds. This bond that goes to this carbon and this bond that comes to this other carbon. So it is in between two carbon atoms. It is called a secondary amino group, which is called an amino group. Now you can see this one is valine. This is valine. Valine is just like when you have a propyl group attached. Just like when you have a propyl group attached to that carbon, call it valine. Now you can see that the structure of valine is similar to the structure of leucine. And you can see this one is valine. This is valine. Valine is just like when you can see this core cycling now is the same thing that is found in valine. Just the only difference is that there is an extension of CH2. So if you extend CH2 and extend the propyl group, you have leucine. So valine and leucine are very similar. If you can draw the structure of valine, you can actually draw the structure of leucine. Then leucine and isoleucine are almost also similar. It's just rearrangement of carbon atoms. It's just rearrangement of carbon atoms. You can see that the number of carbon here are still the same. We have 1, 2, 3, 4. 4 carbon atoms. How many hydrogen? 1 plus 3 here, 4, 6, 9. The same thing we have here. We have 1, 2, 3, 4. Then we have 9 hydrogen. So they are similar. That's why we call it iso. Leucine and isoleucine. So it's just a spatial arrangement of the atoms in space that differentiate leucine from isoleucine. Now we have the methionine. The methionine are example of sulfur-containing amino acid. There are 2 sulfur-containing amino acids, methionine and cysteine. Now, for the lady that asks, how do we differentiate polar amino acid and non-polar amino acid? Now, non-polar amino acid, you can see that it will not have any polar group. Polar group that we know is OH. OH is a polar group. OH is a polar group. C, O, OH, or COO minus is a polar group, which is a carboxylic group. This is the alcohol, OH, this carboxylic group. Nitrogen, amino group, NH2 is a polar group. NH2 is a polar group. We have a thiol group, which is a sulfur group. SH is also a polar group. So any amino acid that has OH, COH, NH2, SH are polar amino acid. They will interact well with water. Now, listen to the statement. If these amino acids are found, if this non-polar aliphatic arrow group amino acids are found in the protein, they are usually found within the core of the protein or in the part of the protein that is embedded within the membrane bilayer if the protein is an integral protein. I wish that we'll have through a lecture series where I'll break down core topics in biochemistry so that we can actually have a grasp of what biochemistry is talking about. So why are they found within the core of a protein? Why are they found in the core of the protein? Now, the core of the protein is usually a part of the protein that is isolated from the aqueous environment. You know, most proteins are usually in a fluid or in a liquid. And that liquid is usually water, which is hydrophilicity. So a molecule, a part of the protein that will be interacting with the water should be hydrophilic. So if you put the protein into water, the protein will fold such a way that it will sequester or it will prevent the hydrophobic part of the protein from interacting with the water. Or we know that because they are hydrophobic, they will try to shy away from the water, then allowing the parts of the protein that are hydrophilic to interact with the water environment. So if these proteins are found, if these amino acids are found in the protein, they will try as much as possible to shy away from the water environment. In doing so, it will result in a particular structure of the protein. So if this protein, if this amino acid are found in the protein, they will try as much as possible to shy away from the water environment. In doing so, it will result in a particular structure I'm trying to see if I can actually work on the audio. Trying to see if I can actually work on the audio. I think because it is actually linked to YouTube, or I'm thinking of something. Trying to see if I can actually work on the audio. Trying to see if I can actually work on the audio. Actually linked to YouTube, or I'm thinking of something. Mr. Matt, have I answered your question? Mr. Matt, have I answered your question? So I want you guys to be asking your questions on the chat column, but I can carry everybody along. So I want you guys to be asking your questions on the chat column, but I can carry everybody along. I want you guys to be asking questions on the chat column, but I can carry everybody along. Okay, somebody asked me what are the example of nonpolar amino acids. I hope I've solved the problem of video echoing, audio echoing. I think so. I've done that. So let's progress. I don't know if people are subscribed. Please do me the favor to subscribe and click on the like button. I've seen only 0 likes. Why we have 36 person watching live. I should be having up to 36 likes. Please click on the like button. So we're progressing. We're progressing. Somebody asks examples of nonpolar amino acid. These are the examples of nonpolar amino acid: glycine, alanine, proline, valine, leucine, and methionine. These are examples of nonpolar amino acid. Now we have the aromatic amino acids. Now the aromatic amino acids are relatively nonpolar. When we say relatively nonpolar, when we say relative nonpolar, we didn't call them nonpolar in full terms. Just like it is relative, and the relative because it might contain some hydrophilic properties or hydrophilic molecules that will make them hydrophilic. So you can see for thyroxine, thyroxine has a hydroxy group. In fact, we call this hydroxyphenylalanine, otherwise known as a thyroxine. We have the tryptophan. The tryptophan, you can see the NH group here, which makes it which confers a little hydrophilicity to these molecules. So they are aromatic because they have a benzene ring attached to them. Now we have they absorb light strongly at 280 nanometer. So when we say they absorb light 280 nanometer, this is one of the reasons why we use this particular wavelength in quantification of proteins. If we want to know if the protein is present in a solution, we set the absorbance at 280 nanometers. So if there's absorption of light within that wavelength, we know that there's presence of protein in that solution. So you might have a solution, you want to test if there's presence of protein in that solution or not. And because of most protein, we have 1 or 2 of these aromatic amino acid, they will absorb light at that wavelength. So if they absorb light at that wavelength, you can conclude that yes, there is presence of what? Protein in that solution. Now, thyroxine kinases or tyrosine and tryptophan are slightly hydrophilic because of the hydroxy group and the amino group. Now, when we say thyroxine kinases, these thyroxine kinases are enzymes that phosphorylate a protein at the thyroxine residue. And it is because of the hydroxy group that enables that protein or that enzyme to be able to phosphorylate thyroxine because of the hydroxy group in thyroxine. Remember, the phosphate group is just like a carboxylic group. It will react with the hydroxy group to form ester. So we call this kind of ester, we call them phosphate esters. So it will react, it will phosphorylate the protein at the thyroxine group, making the activating or inactivating the protein. Because phosphorylation can activate or deactivate a protein. I wish we'll convert this to a lecture series where we look at even metabolic pathways and metabolic regulations. But let me not dwell. These are aromatic and they absorb light strongly at 280 nanometer. So in case if you asked, which of these amino acids absorb light strongly at 280 nanometer, you should be expecting phenylalanine, thyroxine, or tryptophan in the options. Sometimes you see phenylalanine, you also see option A, phenylalanine, option B, thyroxine. I will be on some D, but I might take glutamate or some D, alanine. Now, because you have two options, you are supposed to select the two options. Unless the system doesn't allow you to select two options. But if the system allows you to select two options, select the phenylalanine and thyroxine. Failure to do that, you're failing the equation. So this other classification is based on positively charged or basic arrow groups. Now, these amino acids are positively charged. Example is lysine, arginine, and histidine. Then we have negatively charged or acidic arrow groups, which is aspartate and glutamate. Now, this positively charged and negatively charged amino acid, they are all polar. They are all polar. They can interact with water because of their charge groups. They are all polar. So note that they are all polar, but this one are polar but positively charged. These are also polar but negatively charged. So the only two negatively charged amino acids are aspartate and glutamate. Now, looking at the structure, it is very easy to draw the structure of aspartate and glutamate. If you look at aspartate, we have CH2COOH. In glutamate, it's just an extension of additional CH2, just an extension. So if you can draw aspartate, invariably, you can also draw the structure of glutamate. So we have the polar uncharged arrow group. This one, they are polar, but they are not charged. The previous one I just showed you now are polar but charged, either positively charged or negatively charged. Now the polar uncharged arrow group means they don't have any charge. You can see, previously when Miss Cynthia was asking me, how do we differentiate polar and non-polar? So you can see for these molecules here, serine has hydroxy group. It is polar. Threonine has an hydroxy group. It is polar. You can see that the structure of serine and threonine are almost similar. The only difference is extension of what? CH3. So if you can draw the structure of serine, just extend it with CH3, you get the structure of threonine. The same thing goes for cysteine. If you can draw the structure of serine, you can draw the structure of cysteine. Instead of putting OH in serine, you put SH, you get cysteine. Now remember in our previous one about the negatively charged amino group, which is aspartate and glutamate. The asparagine and glutamine derive their name from aspartate and glutamate. For aspartate, for asparagine, looks like aspartate. The only difference is that where you have COO minus in aspartate, you now have instead of the oxygen here, like aspartate, you have amino group that makes it asparagine. For glutamate, you have oxygen here only, but for glutamine, the oxygen is replaced with what? Amino group. So we call it a glutamine. So if you can draw the structure of aspartate, you can draw the structure of asparagine. If you can draw the structure of glutamate, you can draw the structure of what? Glutamine. It's just a slight difference. So we have these are uncommon amino acids. These are uncommon amino acids. We have hydroxyproline, 4-hydroxyproline, which is the derivative of proline found in plant cell wall proteins, plant cell wall proteins, and what? Collagen. I know you must have heard that. You must have heard about collagen. Collagen are connective protein. It serves as, if you heard about the tendons and ligaments, they are usually collagen. Even your skin is a function of the collagen. Even your gum is a function of collagen. Now, most of you must have heard the medical condition scurvy, which is lack of vitamin C. And lack of vitamin C causes bleeding gums. It causes bleeding gums. I'm coming. I want to show you something. It causes bleeding gums. Let me, I want to see how we fare. Yes, let me, an example of non-polar amino acids. Favor, I'll get to that. Okay, I've given you that answer already. Which other question? Wisdom Boniface, I just did come. We actually set the time. We can't keep waiting. But the video will still be available on YouTube. You can actually replay the video and watch at your convenience. So I want to be seeing question. I said people are not liking the video, people are not liking the lecture. Nobody's responding. Please like. Please like. If I let's take 1 minute to like the lecture. Give me a thumbs up. Give me a thumbs up. I want to be seeing more thumbs up, more likes. And please, if you have not subscribed, try as much as possible to subscribe. And if you want us to continue this as a lecture series, also indicate. So these are common amino acids. We have the hydroxyproline. We have the hydroxyproline, which are found in collagen. I was talking about scurvy, which is a lack of vitamin C. Vitamin C is essential for the hydroxylation of proline. So the enzyme proline hydroxylase is an enzyme that hydroxylates proline. And without the hydroxylation of proline, proline cannot form the fiber that is called collagen. So without proper synthesis of that fiber called collagen, as a result of the deficiency or lack of the nutrient vitamin C, can lead to fragile collagen. When you say something is a fiber, we define fiber based on strength. If the people in engineering will call it strength of a material. If the collagen is not properly synthesized, the strength of the collagen will be weakened. So any little injury, any little contact can cause injury to the system. Just like when you're brushing your teeth. When you're brushing your teeth, you know that the brush will be going through your gums too. So if you have a weakened collagen, you find yourself bleeding while you're brushing. So if you're bleeding while you're brushing, know that you should up your vitamin C intake. You should up your vitamin C intake. So now so that is why vitamin C is important. Vitamin C is important in the synthesis of hydroxyproline and hydroxylysine. Hydroxyproline and hydroxylysine are two major amino acids that are found in collagen. So if you lack vitamin C, the enzyme proline hydroxylase and lysine hydroxylase will not be able to hydroxylate proline and lysine. And failure to do that, the synthesis of collagen will be impaired. And once that happens, the collagen structure strength will be reduced. Therefore, you'll be having fragile arteries, bleeding gum, easy breaking of your skin. So, Edward, one person say, which level is this? This is general biochemistry. It is irrespective of level. It's irrespective of level. So we have 6N-methyl lysine, which is found in myosin. Myosin is actually a muscle protein, which is involved in contractile, is a contractile muscle protein. So your muscle protein contains a 6N-methyl lysine. So that means the methyl group is attached to the nitrogen group, the cis nitrogen group of lysine. We have gamma carboxyl glutamate, which is found in prothrombin or blood clotting, which is also found in calcium binding proteins like amodulin. Just calcium binding protein. We have the desmethane, is a complex derivative of 4 lysine residues. These are just some common amino acid. We also have selenocysteine, which is a rare amino acid introduced during protein synthesis, not post modification. And when we say not post modification, it means that this particular amino acid is coded by the gene. No, we know that there are only 20 amino acids that are coded by the DNA or by the gene, but this one is also coded by the gene. So it is not hydroxyproline is post transcriptional translational modification. Hydroxylysine is also post transcriptional post translational modification. But for selenocysteine, it is not post modification. So derived from serine contains what? Selenium instead of what? Sulfur. It is actually found in few proteins. So these are uncommon amino acids. So these are essential and non-essential amino acids. So if your diet is rife, what I mean by rife, if your diet is mainly composed of a non-essential amino acid, it means that you're not feeding well. So somebody might be eating, but might not be eating very well. When I was in final year, somebody presented, I think no, that was during my master's. Somebody presented a topic on hidden hunger. Your stomach might be filled, but you're still hungry. So what is that thing that you need that is not present in the bulky food that you've already ate? So those are usually those micronutrients that are necessary for physiological response, physiological activities. So the essential and the non-essential. So we have the histidine. You can see this the superscript A. The superscript A tells you it is a conditional. It's actually something like it's essential for some persons, like for infants. It's an essential amino acid. Like you can see, histidine was once thought to be essential only for what? Infants. So adults can produce histidine, but infants cannot produce histidine. It is now known that small amounts are also needed for adults. So it is no longer only for infants. As an adult, you also need small amounts in your diet for histidine. It means that your body cannot produce the enough histidine that is necessary for your biochemical activities. Now, the ones that are labeled with B superscripts are amino acids that are conditionally essential. When we say they are conditionally essential, it means there are some conditions we have to tag them as essential amino acids. So look at they say if they are either inadequate precursor. Precursor is what is actually used to produce them, or inadequate enzymes available to create these in the body. So sometimes somebody might be sick, or somebody might have a genetic disorder that will render those amino acids essential. For example, thyroxine is non-essential amino acid, but it can be essential if the individual have phenylketonuria. Phenylketonuria is actually a genetic condition that results in excess production of phenylketones. And those phenylketones can be seen in urine. That's what we call them phenylketonuria. And the enzyme that converts phenylalanine to thyroxine is called phenylalanine hydroxylase. If the enzyme is deficient, thyroxine cannot be produced, which will lead to accumulation of phenylketones in the system. So proline, glycine, glutamine, even cysteine and arginine can be conditionally essential in some cases. So now let's look at proteins. Let's look at proteins. I wish we have a lecture series on protein. Protein is very vast. Protein is very vast. The chemistry, the folding of proteins. I wish we can actually have a lecture series on proteins. Now, proteins are large, complex biomolecules made up of chains of smaller units called amino acids, which are linked together by peptide bonds. They are essential macromolecules in all living organisms and play diverse functional roles in the biological system. So proteins are enormous. They function in diverse manners in the biological system. Their roles are so much. They are the major functional parts of the biological system. So you can see on the right-hand part, you can see the structure of a protein. This protein is actually albumin. Protein is actually albumin. So proteins can play structural role in terms of collagen, like I explained to you, the connective tissue or the keratin, which is present in your hairs and nails. They can play enzymatic roles. They can catalyze transformation of biomolecules from one form to the other. You can see they can actually look at a molecule like glucose and break down glucose so that glucose can give you energy. Let me put it in the simple form just like that. So they can also serve as transport system where they carry molecules from one region, from one part of the body to another part of the body. Example is oxygen. Hemoglobin is actually a protein that transports oxygen from the lungs to various parts of the system. We also have, they can also form as defense system, like the immunoglobulins. The immunoglobulins are proteins that fight against foreign bodies. So we have signaling. So they can actually form as signals, telling the system or showing the system how the system can respond to external influence. For example, we have insulin, like we have hormones like insulin. Insulin can actually tell yourself there is glucose available. Take in glucose so that you can actually have energy. We have, okay, let's look at this, how proteins are formed. The peptide bond. Now you can see this amino acid 1, you can see the amino acid 2. Now the amino acid 1, you can see it here. You can see the amino acid. This is one of the amino acid. This is another amino acid. So remember, we are not looking at the arrow group now because we are assuming the arrow group to be any of the amino acids. So this can be any amino acid. It can be glutamate, it can be aspartate, it can be arginine, it can be leucine, it can be isoleucine, it can be glycine, any of the amino acid. Now, the hydroxy group is removed from one of the amino acid from the carboxylic end, while we remove hydrogen from the other amino end of the second of the other amino acid. So if we remove this hydroxy group and hydrogen, will produce water. After producing water, the two amino acids will now be joined together You can see them here joined together to form this bond there is called the peptide bond. It's called the what? Peptide bond. And in tests for protein also, for we to know that there's protein in a solution, we can use a test that can detect the peptide bonds in protein. And such tests like biuret test. The biuret test can detect the presence of that peptide bond. So once we introduce a solution into a biuret solution, or we introduce a biuret solution into biuret reagent into a solution that we don't know if the solution contains a protein, it will change the coloration of the biuret reagent to purple. Once that happens, you know that there is presence of protein. So if you have a solution maybe that is colorless, you don't know that there's protein in that solution. If you add a biuret reagent, it will turn the blue biuret reagent into purple, telling you that yes, there is presence of what? Protein. If there is no change in the color from blue to purple, it means that there is absence of what? Protein. So we have levels of protein organization. There are 4 levels of protein organization. So if you ask, which of these is the level of protein organization, or name the different levels of protein organization. You have the primary structure, you have the secondary structure, you have the tertiary structure, and the quaternary structure. Now let me explain. You can see the primary structure here. The primary structure is just a linear sequence of the amino acid. A is one amino acid, B, C, D, E represent different amino acids. Now, if these amino acids that are in the primary form start interacting gradually with each other, in order they start forming either helices or beta sheets, we refer those kind of those level as what? Secondary structure. So usually, when there is interaction between the amino acid 4 residues apart, it will form a alpha helical structure, either right-handed alpha helix or left-handed alpha helix. Therefore, for beta sheet to be formed, there's usually a steric hindrance in a particular amino acid, and those particular amino acids that causes beta sheet to be formed are glycine and proline. Glycine and proline are 2 amino acids that can cause a beta turn. I wish we go into protein structures. We're going to protein chemistry properly. So they introduce there's what we call type 1 and type 2 beta turns. So if you have a glycine or proline, they can cause a steric hindrance that will make the protein to start forming like a sheet, a bar, looks like a sheet of bar, like a sheet of chocolate, a bar of chocolate. So it will form like a bar. We call it a sheet. So we call it the beta sheet. Then tertiary structure is when we have more than one either alpha helix or beta sheets. So alpha helix and beta sheets both of them can be found in one protein. These 2 secondary structures can be found in one protein. And if they are found in one protein, they can be called tertiary structure of protein. So tertiary structure are usually formed between more than one secondary structures. So there are some tertiary structures that will be entirely like what you're seeing here. What you're seeing here is albumin. What you're seeing here is albumin. Or let's just assume it's actually a protein. So what you're seeing here, you can see there are just only alpha helix, more than one alpha helix. So this is a tertiary structure. It's a tertiary structure. So in some protein, you also see only beta sheets. More than one beta sheet is also a tertiary structure. But in some other proteins, you can see some proteins contain both alpha sheets and beta sheets. Those are also secondary, those are also tertiary structure of proteins. We can also have a quaternary structure of protein. In quaternary structure, we look at two tertiary structures joining together to form quaternary structure. So quaternary structure is more than one tertiary structure form the quaternary structure of proteins. So you can see two tertiary structure joined together to form a quaternary structure. You can see two or more. Like hemoglobin has two alpha sheets, two alpha subunits, and two beta subunits. So total of 4 subunits that makes up the hemoglobin. So the hemoglobin is the tetramer. When we tetramer means that it has three protomers or three, I say three, four rather. We say tetramer. That's 4 subunits linked together. So it is called the quaternary structure. And sometimes there might be interaction between the subunits. So that is what we'll get when we get into cooperativity. For those of us that wish to go into protein structures or protein chemistry. So if the individual subunits are interacting with each other, we call it cooperativity. So the cooperative can be a positive cooperativity or negative cooperativity. Sometimes the individual subunit might not also be interacting. If they are not interacting with each other, we'll say there is no cooperativity between the subunits of that protein. When we get into protein chemistry and the kinetics of multi-substrate or multi-subunit like hemoglobin, can now start looking at into their cooperativity, their kinetic plots, how they behave. Thank you. So I'll be expecting more questions at this point. So use the chat column. Use the chat column on the YouTube to ask your questions so that I can be able to respond to your questions. Okay. Thank you, Prince Matthew. We have just stayed there for almost 1 hour, 19 minutes, and we are done. Of course, the video will be downloadable. The video will be downloadable. So please, I want you to ask your questions so that we can actually, I can actually attend to them. And probably, if you want us to fix another lecture between now and today is Thursday, between now either Sunday evening by the same time, just indicate so that we can actually have another lecture. Maybe that lecture will just prepare you for the Monday test. I'm not still getting likes. I don't know why I'm not getting likes. What is the Saturday will be good. Okay, I'll post a link so that we can actually. I don't know if we should have. I don't know if we should have a WhatsApp group where we can be posting this link so that we can actually have easy communication. I can post the scheduled lecture time so everybody can sit at the center. We will not have to depend on course reps or people to propagate the information. So in that WhatsApp platform, we'll do a poll to know the day that will be better. I'm expecting questions. I'm expecting questions. Please throw in your questions. I'm expecting questions. Okay, let me, I think I have to create let me create the WhatsApp link so that I can post it. Okay, somebody said I should explain the tertiary level. Okay, for the tertiary level is where you have more than one secondary structure. So if you have two secondary structures linked together, it is called a tertiary structure. So the two secondary structure might be two alpha helix or two beta sheets, or one alpha helix and one beta sheet joined together, or three alpha helix, three beta sheets joined together. They are all secondary structure, they are all tertiary structure. Okay, I posted the link to the WhatsApp group, so you can join the WhatsApp group. Okay, you asked I should explain the building block of protein. The building blocks of protein is just the amino acids are the building blocks. The amino acids are the building blocks. For example, if you have alanine attached to glutamic acid, it becomes two, two amino acids linked together by a peptide bond. That is actually called a dipeptide. If you have three joining together, three amino acids joining together by a peptide bond, it is called a tripeptide. If you have four, it is called tetrapeptide or oligopeptide. If you have 5, 6, 7, 8, 9, 10 to 50, it becomes a polypeptide, which is a protein. So the building blocks are the amino acid. Those are the amino acids that makes up the protein. And the overall structure of the protein is determined by the sequence of the individual amino acids that are found in that protein. Ernest, you asked the quaternary structure. I said the quaternary structure joined together. You have two separate tertiary structures joined together is called a quaternary structure. For example, more than two, right? More than two, like two or more. Let me put it like two or more, or let's say more than one. More than one, they are now referred to as each of the units is now called a subunit. Each of the units is now called a subunit. So we have, for example, hemoglobin. Hemoglobin has 4 subunits. So that means it has 4 tertiary structure joined together to make it hemoglobin. So hemoglobin is a quaternary protein made up of 4 tertiary proteins. Do you understand it, Ernest? My dog, you there? Madam, there. People no book pass me. I beg you. Thank God for having her here too. It's not easy having a scholar. Okay. Lina asks, my confusion is around amino acid under non-polar amino acid. I said non-polar amino acids are those amino acids that doesn't like water. They don't like water. So they are usually referred to as hydrophobic. They have phobia for water. And if you look at their structure, you find out that you won't see anything like hydroxyl group attached to them, carboxyl group attached to them, amino group attached to them, thio group, which is SH, attached to them. So if you don't see any of this polar functional group attached to them, it means they are hydrophobic. Lina, do you understand what I just said now? Lina, do you understand what I just said now? Okay, so the video will be available on YouTube. You can revisit the video and watch again. Any other question? So click on the link. Click on the link. Click on the link to join the WhatsApp platform. Cynthia, this lecture is not for DNA. Just to say something about DNA. Well, we'll definitely get into DNA, nucleic acids, and nucleotides. But I don't know what you want me to say. There are a lot of things to say in DNA. I don't know where your question is actually going. Do you want me to start talking about DNA? say an RNA. Yeah, okay. Wait. And did you just ask this question about proline? I've seen a question on proline. I said proline is an amino acid. Others are known as amino acid. Proline is an amino acid because the amino group is a secondary amino group. It's not the primary amino group. So you look at the structure of proline, you find out that the amino group is attached to 2 carbon atoms. So it's a secondary amino group. So if the amino group is attached to only 1 carbon, it is primary amino group. If it's attached to 2 carbon, it is secondary amino group. So for proline, proline is attached to 2 carbon. The amino group is attached to 2 carbon, so it's called an amino group. So proline is also, the other name we call proline is amino acid instead of amino acid. So I think we should, the other questions should be focused on the WhatsApp, questions should come through the WhatsApp platform and I'll address it there. So let's call it the day. Thank you very much and God bless you all.